Okay, so within my talk, I now would like to bridge the gap between human electrophysiology and animal electrophysiology. In particular, I would like to provide an example where knowledge from combined fMRI and electrophysiology in animals could be used to gain knowledge or can be applied to human electrophysiology in order to gain insights into cortical-subcortical interactions. Similar to my previous speakers, I will concentrate on functional connectivity at rest. And you have heard already a lot about resting state network activity, and you can obtain them, or obtain these networks through calculating correlations across the whole brain. And the nice thing about these resting state networks is that they can be obtained from different studies and from different centers. Moreover, they allow us to study whole brain activity and functional connectivity from only one fMRI scan of 5 to 10 minutes. And in addition, patient populations can usually be easily studied because we don't need a particular task which is matched to the abilities of the patients. And therefore, changes in resting state networks have been detected now for many neurological diseases, and they have been suggested as a potential biomarker. Importantly, I think, by now these resting state networks have also been identified through MEG. Despite all these advantages, there are of course limitations, and they have been touched already before. One of them, which I will talk about, is that the time resolution of fMRI is usually not in the order of our actual neural activity. The first solution you might think of is to use EEG or MEG. However, most studies using these techniques have so far been calculating the functional connectivity also over a longer time range. The reason for this is that they tried to mimic the results from fMRI, taking them as gold standard. More recently, researchers have started to estimate time-resolved network changes, and the study by Baker et al. Showed that the state lifetime of a network is in the range of 100 to 200 milliseconds. We actually hypothesized that even faster events are responsible for the network generation, so-called neural events. And this hypothesis was based on a study by Logothetis and colleagues, where they recorded electrophysiology and fMRI in primates. They recorded electrophysiological activity within the hippocampus and then afterwards detected sharp wave ripples. Ripples are associated with the retention of awake experiences and memory retrieval. And until this study, it was not clear how these hippocampal ripples relate to whole brain activity. And what Logothetis and colleagues did is they took these hippocampal ripples as neural events and averaged the bold time series across the whole brain. And through this, they obtained a map of activations and deactivations related to these hippocampal ripples. And they found that most cortical areas are actually activated, while subcortical areas are deactivated. And this speaks for a preferred communication between hippocampus and cortex during these ripples, while the communication to the subcortical areas is silenced. And that could in part explain how hippocampal ripples are related to memory retrieval. We went actually a step further and said, well, it's probably not only the hippocampal ripples which are relevant for network generation, but every neural event could potentially be related to network generation. And therefore, you see here these stylized neural events, and below you see the bold activity recorded. And what is commonly done is that you compute the bold activity, the network across the whole bold activity. However, if you would be able to identify these typical these typical events, you would be able to determine subnetworks. And these subnetworks are, of course, contained within this whole bold connectivity network, but by only recording the fMRI bold activity, you will not be able to extract these subnetworks. So far, this approach of net fMRI has only been used in animals. And the question is, of course, can we do a similar approach in human data? Until now, a reliable estimate of subcortical electrophysiological activity in humans is only possible through invasive recordings, because most EEG and MEG techniques are restricted to the cortical areas. Therefore, we are limited to patient data, and in particular, we can look at recordings during epilepsy surgery or at the implantation of deep brain stimulation electrodes. Still, due to safety concerns, we cannot combine these recordings with fMRI in humans, and therefore a potential solution is to combine it with EEG or MEG. In my talk, I will concentrate on deep brain stimulation in Parkinson's disease. In this case, electrodes are stereotactically implanted into the subthalamic nucleus, a basal ganglia structure. Through applying high-frequency stimulation to the subthalamic nucleus, the motor symptoms of these patients can be greatly improved. However, so far, the exact mechanisms of deep brain stimulation are not known. Still, we think that by using these surgeries to record from the subthalamic nucleus and combine these recordings with MEG, we can gain insights into the pathophysiology of Parkinson's disease and potentially also into the working of deep brain stimulation. One of the first studies which used this approach was by Jan Hirschmann and colleagues. They recorded 9 Parkinsonian patients for 5 minutes at rest in the MEG. And they calculated the functional connectivity between the subthalamic nucleus and all cortical areas. And what they found was that the coherence in the beta range was mainly between subthalamic nucleus and sensory motor areas. Interestingly, the cortical extent in the high beta range was larger than in the low beta range. Still, in this study, they did not answer any question related to deep brain stimulation, and this was done by a study from Oswald and colleagues. They again used a similar approach by recording Parkinson patients in the MEG after the surgery and simultaneously recording local field potentials within the subthalamic nucleus. Then afterwards, they calculated the coherence between the subthalamic nucleus and the cortex. And in yellow, you see the regions where there was coherence in the beta range. In green overlaid are those regions where the beta coherence in the high beta range was larger than in the lower beta range. And importantly, the red area indicates areas where when you turn off the deep brain stimulator, the beta coherence was reduced. However, this change in functional connectivity actually did not relate to the motor improvement they saw in these patients. The motor improvement was only related to a reduced beta activity within the subthalamic nucleus itself. Therefore, this study still could not answer the question what changes or what functional changes deep brain stimulation produces so that we see these motor improvements. The question then is beta maybe not all we need to know, but maybe there are other frequency components relevant in Parkinson's disease. And by now, several studies have shown that within the subthalamic nucleus, there are also high-frequency oscillations. And in particular, at around 250 hertz, we have a peak in the medication off state. And this is shifted to 350 hertz when the patients receive their usual medication. the question for us was if this is maybe a potential, or if these are potential neural events that could generate global networks. For this, we recorded 9 Parkinson patients, again without their medication, a day after they got their electrodes implanted. At this stage, we could still record simultaneously the local field potentials within the subthalamic nucleus and the EMEG. We recorded on average 15 to 30 minutes of rest with eyes open. And for the later analysis, we selected then from those electrodes the contact which showed the best clinical effect. We then determined on these LFPs high frequency events, and we defined those as an amplitude increase of more than 4 standard deviations above the average within both subthalamic nucleus. We detected events in the range, or tested for events in the range from 100 to 350 Hertz, and the MEG data itself we cleaned from artifacts, of course, and we did a source reconstruction with a weighted minimum norm estimate. We then calculated event-related fields for those detected neural events across the cortical surface. Overall, we only detected across all subjects events in the frequency range from 150 to 350 Hertz. We found an average number of events around 500, with the exception for the frequency range from 250 to 300 Hertz, where we detected 1,500 events. The average duration of these events was in the range from 26 to 43 milliseconds. When we now look, When we now look at the cortical activation related to events in the 150 to 200 Hertz range, we see a clear activation already within the first 10 milliseconds in frontal areas, and at around 20 milliseconds we see a deactivation in the left SMA and then an activation in the right SMA. When we go to events from 200 to 250 Hertz, we again see at around 10 milliseconds after the neural event an activation in the left primary motor regions. For events above 250 Hertz, we did not detect any clear activation pattern. Still, I think, at least for these lower frequency events, we could detect clear activation patterns which could relate to the motor involvement of the subthalamic nucleus. Therefore, I would like to suggest that net MEG could be a method for identifying whole brain activations. The advantage compared to traditional functional connectivity measures is that we can also consider faster neural events, and we can actually look at the time evolution of cortical activations in the millisecond range. However, a word of caution is also necessary. It does not indicate any causal relationship between the neural events and the whole brain activity. We only know that a neural event happened, and time locked to it, the cortical activation is happening. So it's only a temporal relation between those. I have shown you that for the Parkinsonian case, the relevant motor areas and frontal areas were activated based on the identified events in the subthalamic nucleus. It could be therefore a useful method to study whole brain analysis with MEG, and it can complement standard functional connectivity measures. For looking at combined invasive electrophysiology and MEG or EEG, we usually have as methods coherence, correlation, or Granger-based methods. And I have shown you for coherence that in the Parkinsonian case, we can actually well identify functional connectivity. They all have a clear interpretation within their framework. And I hope I could convince you that an inclusion of invasive electrophysiology can provide important further insights into diseases, and it might reduce methodological barriers we usually have in resting state functional connectivity analysis. With this, I'd like to thank my group and my collaborators, and I would like to thank you for your attention.